1984, if I'm not mistaken. So it's a long time of work. And during this time, she worked on neurobiology of circadian rhythm, and she did her PhD and master's in this field. And she also headed the unit of neuroscience research on biological rhythms in the university between 1997 and 2017. So, and she was one of the early presidents of the Society for Neuroscientists of Africa, which was between 2011 and 2013, and also between 2013 and 2017. So she's also a member, so it's a lot of things to say. She's also a member of the African Regional Committee of the International Brain Research Organization. And she's also involved in other things that are also in the Arab world, because she's also in the board of the International Society for Neurochemistry, representing the committee for the Arab states and a committee also in the UNESCO. So she's very much internationally renowned. And in order to also promote neuroscience education, she's also involved in the setting up of 2 neuroscience networks. One is the one in Morocco, which is between the GDRI in Morocco, which is between Morocco and France. And the second is the one in Europe itself. So, and in terms of publications and what she's been doing to advance our publication from Africa, she's a reviewer for neuroscience letters so at the moment, and she also reviews for Frontiers in Behavioral Neuroscience and a bunch of other journals. So it's a pleasure, and I'm out to anchor to this program. So I leave you to Professor Nuria, and I'm sure you will enjoy the talk so far. So Professor, you can take the stage. Thank you so much for accepting our invite.
Okay, thank you very much, Abdul Basit.
You're welcome.
It is an honor for me to add something to this webinar series by presenting a part of the work that we did in our laboratory in Rabat in collaboration with Professor and Dr. Hamid Ben Azzouz in Bordeaux. So my affiliation is the Mohammed V University. I retired for many years now, but I continue to work with the IBRO Africa Center for Advanced Training in Neuroscience, as well as with one of my students on this topic of research, neurotoxicity and Parkinsonism. In 1997, I created, just after my state doctorate, a research unit on biological rhythms and environment. And the first domain of research was on endemic species, the Gerboajaculus orientalis, in which we tried to understand the mechanism of photoperiod integration by the brain and the control of the photoperiod on reproductive function. So this topic continued to run in the laboratory with Dr. and Professor Nezha Bouhedou, who was one of my PhD students. But my talk today will concern the second topic, which is neurotoxicity of environmental pollutants, focusing on Parkinson and Parkinsonism, and I will explain later why we have chosen this neurodegenerative disease. And we have also focused on metals and the heavy metals like manganese and copper, because they are elements of physiology of the body, and heavy metals like lead, which is not localized in our body and enter the body from the environment. We have also studied the consequences of these metals on the brain, particularly by measuring the motor and non-motor functions. And after that, we have tried to demonstrate that pineal hormones, melatonin and 5-MeO, can have a prophylactic effect, and this will be will not be exposed today. And we have also tested paraquat and maneb toxicity, and now we work on the suprachiasmatic functions and circadian rhythmicity in rat model of Parkinson, particularly in cis-OHDA model of Parkinson. So the purpose of this SONA webinar lecture, before starting, I want to congratulate the young people and neuroscientists of Africa who has taken in charge this webinar series, which permits to us all to share our knowledge in neuroscience and permit to young people to understand many things concerning neurodegenerative diseases and other topics. And I would like also to congratulate our Secretary General, who did with these young people a very nice work in SONA. So thanks for SONA, thanks for all the organisms that promote and fund these activities. So I will introduce my talk by explaining why we have chosen to work on neurotoxicity and PD. And after that, I will present the most important results we obtained and published concerning lead intoxication and the toxicity, and also the non-motor deficits, the non-motor functions that are linked to lead toxicity by presenting all the works done on circadian rhythms, hoping that this can be can permit to us to understand the problem of sleep-wake cycle in this disease. So all the work have been founded by many agencies here. If someone is interested to understand what are these agencies, after the end of the talk, I can present and explain. So now more and more studies started to make and to run with epidemiological studies concerning Parkinson's disease in the world, and particularly in Africa. In Africa, we have not, during many years, any information concerning epidemiological parameters of this disease. But now we can find in the web and in the World Health Organization many informations showing that in Africa, we have now a prevalence rate that increase in sub-Saharan Africa between 30 to 60 cases per 100,000 population. And in the north of Africa, the prevalence is more higher than in sub-Saharan Africa. We don't know exactly why, but I think that the genetic parameters can be introduced at this level. And you know, we know all that this disease has a very high cost over the world, but in Africa, the cost is principally a cost of the family that take in charge the patient more than the institution, etc., because it is low or middle income country. After that, I can say that more and more patients dead with the Parkinson disease because the medicine or the deep brain stimulation or a lot of things are of a very high cost, and many population, a lot of persons that suffer from this disease have no access of medicine and hospitals. And now there is more and more projects that try to understand the causes of this disease in Africa by running a project, and this can be found in the Michael Fox Foundation for Parkinson's disease, and particularly these projects are in sub-Saharan Africa, from in Ghana, Nigeria, Sudan, etc. Now, when we see what happens in north of Africa, in which my country is located, we see that we have a growth of Parkinson disease and related movement disorders. And this has permitted to us to give an attention to this disease and try to study the causes and particularly find an additional hallmark of this disease. So for those who don't work on this disease, we know that Parkinson disease have been described very early by James Parkinson as a shaking palsy. And very later, this disease and other persons have discovered that the disease is linked to the loss of dopaminergic neurons situated in the mesencephalic ventral region of the brain in rodent and in the substantia nigra or locus niger in human. Here, you have a brain of normal individual with the substantia nigra containing dopaminergic neurons, which are black because there is a lot of neuromelanin that is a characteristic of these structures. And here you have a brain from a patient who is a Parkinsonian, and you see that there is loss of these neurons that will be responsible for many symptoms. You can see here not a person, but a statute showing a rigidity and the posture of this Parkinsonian patient. So we can see also with using PET scan that we have a very high loss of dopaminergic neurons in the locus niger. And the locus niger here in these figures, in this schema, in this picture, is here in the substantia nigra Pars Compacta in blue. In black, you have the Pars Compacta reticulata, substantia nigra reticulata. And we see that substantia nigra Pars Compacta project particularly in the striatum and in the putamen, and the substantia nigra reticulata in the other region of the brain. So the pathophysiology of the disease concerns the loss of dopaminergic neurons because there is a misfolded protein, alpha-synuclein, which will accumulate in the neurons of the substantia nigra, and then they produce what it is known as Lewi body without O, Lewi body. And these Lewi body are responsible for all the symptoms and are considered as a hallmark of this disease. They are also responsible by dementia in Parkinson patients. I would like to say that Parkinson disease is a very, very hard disease. It develops during a long time. It is a neurodegenerative disease, and there is evidently medical with medicine and also physical like deep brain stimulation that can permit to not if I say to not block the disease, but just to permit to the patients to have a better life in their work. So the interest of our studies concern another hallmark, another marker of the disease, which was developed in the laboratory of Hamid Ben Azzouz in Bordeaux, and all the electrophysiological studies we did have been run in this laboratory. And in addition to the alpha-synuclein, we have the pathology of the disease, which concern the basal ganglia. And in basal ganglia, you have the putamen, the globus pallidus external and internal part. We have here the substantia nigra Pars Compacta and reticulata and another structures. And in these structures, in the basal ganglia network, we have only the subthalamic nucleus, which is of glutamate nature, and which is the structure which stimulates the other structure of basal ganglia, since you see that all the other structures are of GABAergic nature and don't are inhibitory structures. So when we place an electrode in the substantia in the subthalamic nucleus, here you see it, in the subthalamic nucleus, to record the electrical activity of this nucleus in normal in normal individu or in normal animals, you record regular spiking with the regular frequency. And when you do that in Parkinson patient, you have a change from the regular spiking to burst spiking with a very high frequency during very few seconds, which repeated during the time. And this is one of the hallmark of the disease we were interested in. So here you have modelization of the basal ganglia with the substantia nigra Pars Compacta, which project to the caudate and the putamen dopaminergic neurons and release, which will interact with D1 and D2 receptors. I will not specify exactly and spend the time in this, but we have two pathways, direct pathways, which will permit to inhibit the substantia nigra reticulata and globus pallidus internal parts, and then permit the GABA to not be released here, and the thalamus can at this moment release glutamate on the cortex, motor cortex, to regulate the motor coordination, motor, etc. Excuse me. The other pathway is indirect pathway, which leave the caudate putamen to reach the SCN neurons. And we see here that SCN neurons in all the pathway, globus pallidus, substantia nigra, reticulata, and globus pallidus intern, promote and strengthen electrophysiological signals, which are normal in all the structures in normal persons, and become bursty in patients with Parkinson disease. So it is not my specialization, but now I know that, and we work on this, and we know that the motor symptoms of Parkinson disease concern bradykinesia, which is lowering of movements, rigidity, tremor at rest, and postural abnormalities. And we know also that these motor symptoms appears when we have 60 to 70% of progressive loss of dopamine neurons in the locus niger, and then 80–90% decrease of dopamine release in the striatum, who is considered as the enter door of basal ganglia. The other things of Parkinson disease concerns the non-motor symptoms with the neuropsychiatric symptoms, depression, anxiety, cognitive dysfunction, etc. Sleep disorder, problems in autonomic symptoms, gastrointestinal symptoms, sensory exhaustion, fatigue, and other symptoms. So it has been, we don't know at now what are the causes of this disease. We know that between 10 and 15% of the patients get the disease because it is genetic. They get it from genetic version. This disease attain aged person, so the most risk factor is age. I want to say that in Africa, I think that this parameter can be taken in consideration, but we have most of the population that are young. It is not like in Europe or in USA. And it appears that the cause of the Parkinson disease can be a multifactorial disease associating pollutants like pesticides, solvents, and metal with epigenetic and genetic sides. So there is many publications that have proposed and demonstrated that patients with Parkinson disease, like this very interesting paper, patients with Parkinson disease have a high levels of heavy metal like lead. And this paper has definitively concluded that lead exposure is a risk of a PD, at least in USA, because this work has been published from the work done in USA. So, aging, free radicals, oxidative stress. It is okay because free radicals, oxidative stress are linked to aging, but can be also linked to heavy metals accumulation in the body, particularly in bones. You know that lead is a metal that is penetrates the body by olfaction, by cutaneous, and by ingestion of polluted food or water. But in the body, it accumulates in the bones, and during years, it can release it another time in the blood to reach the brain, because he used the calcium channels and also the hemoglobin to be transported from the blood to the brain, because it is with this mechanism that it enters the brain, because there is a blood-brain barrier. So we can now make attention that researchers throughout the world have published many papers concerning this disease or Parkinsonism linked to the toxicity of lead. From these papers, we have all those who presented problems in cognition and emotion, those who presented the presence of alpha-synuclein as a misfolded protein responsible for the death of dopaminergic neurons, but no study has made attention and used electrophysiologic recording of the subthalamic nucleus to add another hallmark of the possibility of lead to induce Parkinson's disease. And very few studies have used and tried to connect sleep disorders and circadian rhythm as symptoms of lead toxicity, and demonstrate that lead toxicity can induce symptoms like those observed and demonstrated in Parkinson patients. So my story with lead and also manganese, copper, etc., and the work we did in our laboratory was emerged from a stay in Ouarzazate, which is in Atlas Mountain, in high Atlas Mountain, and in which there is a lot of mines. And in this, it was a period during which I organized an IBRO school, IBRO ESN school in this town, and the discussion with the physician of the hospital permit to us to understand that many old persons around these mines suffer for Parkinsonism or Parkinson disease, and this has attracted and permit to us to develop this research topic in our country. So neurological disorders in Morocco concern Parkinson, Alzheimer, high level of Saturns in children, which is linked to lead toxicity, a lot of cognitive troubles in low and even in high schools, amyloid sclerosis, a lot of epilepsy as in other countries of Africa. But so schizophrenia, I don't know if this is a real number, but there is a lot of schizophrenia and also a very high incidence of brain cancers. So here we know now that we have every year 4,000 new cases of frequent extrapyramidal symptoms linked to environmental pollutants, particularly in towns around ethologic mines. So the first work concerns the demonstrations that lead intoxication can induce changes in monoamines in cortex or in lateral septum and induce motor and non-motor disabilities. We have in this work chosen to use the subthalamic recording of electrophysiological functions to demonstrate another and to give another hallmarks of this lead-linked pathology. This work has been done entirely by Mariam Sabbar. A part of this work has been run in Rabat and the part in Bordeaux for electrophysiological study. The experimental design is very simple. We have treated with Western rats with lead acetate and the control with sodium acetate for 3 weeks. And after the end of the treatment, we have put the animals in open fields to have the exploratory behavior and the motor behavior, in rotarod apparatus to have motor coordination. We used the sucrose preference to have an idea about perhaps anhedonia in these animals, and then a marker of depression, and plus maze to measure anxiety, and then animals have been put in a stereotaxic apparatus to record by electrophysiology the activity of the SCN. So the first result demonstrates that lead blocks body gate. Here there is 2 different experiments. So the rats cannot gain a body weight in the 2 cases. The explorative behavior in open field test, and here you have an actimeter with the photoelectric cell that permits to record the movement, horizontal and the vertical movements of the animal. And here a sample apparatus that can be constructed by our students when they have not the funds to buy this apparatus. So at all the levels of the cognition and exploration, we have a downing of horizontal movement, downing of vertical movement, and downing of stereotyped movement, which strengthen the hypothesis, at least, that lead interfere with the cognitive and exploratory functions. For measuring motor coordination, we put animals here in a rotarod. Animals, here we have a bar that rotates between 4 and 20 rotation minutes, and we record and measure the time spent on the bar. And we see here that animals with lead intoxication cannot remain longer on the bar when compared to control, which demonstrate that they have a very big problem and very strong problem of motor coordination. After this, we recorded the SCN activity, electrophysiological activity in these animals. And what we see that it is the firing rate in these animals doesn't change between lead intoxicated and controls, which is exactly what happened in cis-OHDA model, rat model of Parkinson. But when we measure the pattern of electrophysiology, firing pattern in control and lead, we find, and we have demonstrated, that we move from regular here pattern to bursty pattern, like what have been published and demonstrated in a rat model of Parkinson's disease. This is a very, very strong parameter that can led us to say that lead can be a risk factor of Parkinson's disease. And here we have immunohistochemistry of cytochrome oxidase, which is more present in the brains of intoxicated animals than in control, which is another marker of the level of the toxicity of lead. The measure of monoamines by high field chromatography, I find this on the web, showed that the only neuroadrenaline in the frontal cortex that is changed, it is lowering, it's decreased between animals in normal condition compared with those who was and were intoxicated by lead. We see that dopamine doesn't change, which was, we were very surprised to see that dopamine and its product of degradation, the DOPAC, doesn't change, as well as serotonin. So this particular result conduct us to the BRAAK theory, which is very, very important when we want to work on this disease. We see here that we have motor disabilities and motor symptoms that are discovered or appears in the patient between 20 and 10 years after non-motor symptoms. And you see here that the patient can start 20 years before by constipation, so troubles in digestive apparatus, but also it is very important to say that problems of olfaction also appears very early. Then, after these symptoms, we find troubles in the sleep wake cycle, and these very early parameters and symptoms are linked to degenerescence first in the early stage of the disease, in the locus ceruleus, which is the part of the brain who synthesizes neuroadrenaline, and also the raphe nucleus, before attaining the temporal, before attaining the substantia nigra in the stage 3, and then all the cortex for all the monoamines. So this theory has permitted to us to understand why the neuroadrenaline change and not dopamine change. And then we have looked to non-motor symptoms to see if these neuroadrenaline changes can be linked to disabilities in non-motor symptoms by recording here the exploratory behavior. And we see, as I said earlier, that we had the motor behavior decrease, but also here the explorative behavior decrease. Here you have an expression of anxiety in the rat recorded in the plus-maze, since these animals don't want to go to the open arms because they are in an anxious state, and remains more close to the closed arms when we put them in the central part of this apparatus. And here we see that we have a decrease in the time spent in open arms as well as the entry in open arms, which is a marker of anxiety in these animals. Concerning sucrose consumption, which give us an idea about the anhedonia of this animal, loss of pleasure to drink sucrosed water. We have put these animals in the cage with 2 bottles. So the paradigm or the experimental manner is explained in the paper, but we have measured the quantity of the water that have been drinking by the animal when the water contain 1% of sucrose and the normal water by weighing the 2 bottles and using this mathematical reaction. So what we have seen and what we have obtained, it is that animals doesn't expresses any depression or any loss of anhedonia. So after that, we have in this unpublished data done by Dunya Salhi, who was a Ph.D. student in the lab, we have tried to link the disabilities observed and recorded in cognitive function, memory and learning, to something that can happen at the level of hippocampus. And we measured the number of the cells reacting to the parvalbumin, since parvalbumin antibodies can be a marker of GABAergic neurons. And we have here the results of this immunocytochemistry of parvalbumin, showing that we have an overexpression in the CA3 of hippocampus, the corn-amon number 3, in this part of the dentate gyrus, as well as in most posterior part, but we remain here in the anterior part of the hippocampus. We need of other experiments to complete this study, which give to us a very interesting results. And I hope that other students throughout Africa can develop the same experimental paradigm to continue to establish the link between cognitive disabilities and something that can appear in the hippocampus. So the conclusion of this first part is that these data provides new evidence, particularly the electrophysiological study for the involvement of lead as a toxicant that can increase the risk factor for having a PD. All the data obtained in this part of the talk are similar to those demonstrated in animal models of Parkinson's disease, or the rat or MPTP, monkey or mouse, and they show that there is a correlation between dentate gyrus overactivity and emotional and cognitive dysfunction. But our experimental conditions doesn't permit to us to correlate lead toxicity with changes or disabilities in dopaminergic system. But it is very interesting to have this positive correlation with neuroadrenaline system that is modulated by lead and may have a significance concerning the early stages of PD, as proposed, as I have explained when I discussed with you a BRAAK theory. So to see if this can be the case, we have moved from non-motor, motor and non-motor symptoms, as I just explain it to another non-motor symptoms, which is of a very, very importance and concerns the sleep-wake cycle. So it is very difficult for us to measure the sleep-wake cycle in the rat because we have not the means to do that, but we have used the activity-rest cycle by measuring the circadian activity of a rat, hoping that the measures can give information that can demonstrate that lead can also be at the origin of perturbation of sleep-wake cycle. So sleep-wake cycle is controlled by many hypothalamic structures, sleep in anterior hypothalamus and wake in the posterior hypothalamus. But the 24-hour expression of the sleep-wake cycle is under the control of the suprachiasmatic nucleus, known as the biological clock that commands all our circadian activity. So we sleep during the night and we wake during the day, and this rhythm is a circadian rhythm. And this rhythm is very disturbed in Parkinsonian patients. Here you have a work published by De Kock in 2008, showing that we have trouble in this cycle with disappearance really of a clear wake stage and the night wake stage. And here we have another patient with a lot of naps during the day, and there is a perturbation in paradoxal sleep. So we have here measured the locomotor activity in nocturnal species, which is the rat, to have an idea about what can happen in sleep-wake cycle, since the SCN is the suprachiasmatic nucleus, is the structure that command the 24-hour expression of these two parameters by loops of clock genes and clock proteins that permit the expression of the circadian sleep-wake cycle. So we have used in our laboratory a very simple experimental apparatus. We put the animals in cages, and we have an infrared captors that capture all the movements of the animals here. We have make a closure between each cage to not have an interaction between each individual. And we have used an ActiveView software to record the locomotor activity or rest activity rhythm. Each line is the record of 1 day. So we have 1 day, 2 days here. And since we have a nocturnal individual and the species, here we have in this line all the duration of the activity during the night for here 14 days. The white places are concerned the rest period, and the black concern the activity period. When we have these records, we put the records in another software to analyze the parameters of the rhythm. This works have been also done by Mariam Sabbar and published in 2017. And the paradigm concern the registration, the recorded of locomotor activity during before the treatment in all animals. And after that, we divided animals. Half was treated by sodium acetate as control, and the other half by lead acetate. This means that each animal is its own control. And we recorded the locomotor activity. Then after we tried to change the photo period of the environment to see if the disturbances in the light-dark cycle of sleep-wake cycle or locomotor activity rhythm can be linked to problems of entraining this activity by lighting, and then we have perfused the animals and see what happens in the suprachiasmatic nucleus. So here we have the double plotting of activity, locomotor activity, circadian activity in the rat control, and here in the lead treated rat before treatment, and here after lead sodium acetate, and here after lead acetate. What we see it is that we have a loss of a part of the quantity of activity during the night in the lead treated animals. Here we see that by Clock Lab software, which give us the activity profile in lead treated animal and here in control animals, and we see clearly that during the day we have more activity in lead treated animal than controls, and also small low activity during the night. Here by analyzing the mean of all the data, we see that lead treated animals have more daytime activity than the controls, which means that there is a fragmentation of the rhythm after lead toxication. Nighttime doesn't change. Here we have the other data that concerns the synchronization by light dark cycle. I return back to my actimeters, which demonstrate that all animals here and all animals here after lead toxication or without lead toxication doesn't change their starting of their activity. So they don't change their activity and don't change the period of the activity. It is only changes in the daytime activity, which explain that there is a problem. So here we see that the synchronization by the light is okay. Animals are very goodly synchronized by light dark cycle. There is an onset variability between individuals in lead and toxicated group. So this also has to be more studied to confirm with a lot with increasing the number of animals. Now we move to another experiment in which we have changed the light on. The first experiment here, the light on is at 6 EM and light off at 20. Then we move to light on at midnight and light off at 2 PM. After that, we move the animals to DD, but I will not present these results. To see if the animals are entrained by the novel photoperiod. So here you have the experiments with 2 animals with lead and toxicity and one control. What we see it is that animals are entrained from starting on their activity at the novel light off, since they are nocturnal animals in all the cases. But if we measure the phase angle, we see that the animals with lead intoxication make more days to synchronize their activity than those in control. So I know that it is very difficult to understand these data when we don't work on circadian rhythms, but we have tried to see in this animal if there is a problem of integration of light. And then we have put animals after 1 hour of darkness under light during the 20 minutes. And we have studied the force induction by light in controls and in lead intoxicated animals. And we see it is very clear. We have a decrease of the force protein induced by light. Is this a problem of accumulation of lead in the retina, or a problem of the transmission of the light from the retina to SCN throughout the hypothalamic tractus? This has to be studied in another experiments later with another. So the chemical organization of the suprachiasmatic nucleus is here. We have the VIP neurons that are located in the ventral and ventrolateral part of the suprachiasmatic nucleus. This neuron is known to be received the photic light information from the retina. And here you have in the dorsomedial part of the suprachiasmatic nucleus, we have vasopressinergic neurons that are known to contain clock genes and clock proteins. So we have studied these two neuropeptides in the suprachiasmatic nucleus of lead intoxicated animals, but before we have measures the optical density of GFAP staining that can give us the possibility to see if astrocytes in the SCN is normal or not in the two conditions. And what we see it is that there is a loss of astrocyte expressing GFAP in the suprachiasmatic nucleus, which can be explained that there is disabilities and the problems of integration of photic information and the loss of force expressed protein after light, which can also indicate that we have processes of degeneration in the suprachiasmatic nucleus in these lead intoxicated animals compared with control animals. It is also the case with vasopressin in dorsomedial parts of the two animal groups, as well as VIP neurons, which now permit to us to move to another marker of the degenerations or the disturbances of suprachiasmatic nucleus activities. And these are, we have considered the clock protein expression since we have a problem in the circadian expression of locomotor activity in these animals. In all cases, we have a veritable decrease of all parameters that are included in the loops of clock, BMAL proteins, as well as PER1 and PER2 proteins, who are very, very important proteins in playing a very crucial role in the expression of circadian rhythmicity, as well as its synchronization by light. CRAN-I2 expression here in this experimental model. So how can be the conclusion of this second part? Several 24-hour locomotor activity parameters were altered in this lead intoxicated animal associated with the content of neuropeptide that are that play a very important role in the integration of photic information, but also for vasopressin, an output of the suprachiasmatic through another structures that control many other functions. The loss of astrocytes may indicate a loss of protection of neurons by astrocytes, and the alteration of clock proteins indicates a decline in circadian expression of rhythmicity. And we can, with this model, suggest that one of the most conserved function in the mammal world, the sleep-wake cycle, can be used as an early parameter hallmark of Parkinson disease and Parkinsonism. So from the first part and the second part of this talk, and on the basis of the results we had, we can conclude that heavy metals are a very serious risk factor, but we need more and more studies, particularly multifactorial studies in which more than one pollutant can be tested. We did that with lead and manganese, and we get a very nice results, but also we have to take in account also the genetic risk factor to have real arguments and to argue that environment has to be cleaned very quickly to block the dispersion of this disease. So I would like to take to thank Sona firstly, and the young people, the young neuroscientists that take in charge these webinars, but also all the members of my lab. The work has been done by Mariam concerning lead electrophysiological part and circadian rhythm part. Dunya has taken in charge the parvalbumin and the GABA part with lead toxicity. Safa Abu Abid worked and got his Ph.D. thesis by working on manganese, and Anas also gets his Ph.D. thesis by working on paraquat and man in our lab. Thank you for Hamid bin Aziz and Fideli possible by opening their lab to our students when we haven't the possibility to run experiments because loss of funds that permit to us to buy equipment. Thank you.